In this training video, we will cover everything you'll need to prepare composites for each of the 3 Nutrition for Precision Health diets. This includes a composite process overview, composite preparation instructions, blending procedures, sample storage, quick notes on the Red Cap Diet Composite production log, and general shipping procedures. Each test diet production kitchen will prepare homogenized samples of each menu day of each test diet according to the schedule found in the composite MOP. These samples will be sent for analytic assessment by a food chemistry testing lab. These results will be reviewed and compared to the NDSR 2022 database planned menus to ensure we are meeting our nutrient targets. These composites will also be used to ensure consistency of the diets over time and between sites. For further detail on any of the topics covered in this video, please refer to the composite MOP. To get started, you'll need a 1-gallon blender, silicone spatulas, distilled water, a wash bottle, a scale that weighs up to 10 kilograms, sample containers, an NSF-approved thermometer, a black permanent marker, clear packing tape, and pipettes, a 60 milliliter syringe, or a turkey baster. Once you're ready to prepare the composite, generate the ProNutra production sheet according to the diet color, the menu day, and the calorie level specified on the homogeneous preparation schedule. Remove anything optional from the production sheet, including coffee, tea, sugar, cream, salt, and pepper. Print the composite production sheet and composite preparation document forms and retrieve the sample IDs from the Red Cap Diet Composite production log. 2 samples will be prepared for each meal day, one for shipment to the food chemistry testing lab and another to store until results are returned. Before starting, label 2 sample containers shown here with a unique ID obtained from the Red Cap Diet Composite production log. The letters A and T will be used to specify the sample that is analyzed and the sample that is temporarily stored. Write the unique ID on the top and the side of each container. Place clear packing tape over the unique ID to ensure it does not get rubbed off or otherwise modified. Document on the production sheets the preparation of each ingredient. Meals used to make homogenate should be prepared exactly as they would be provided to a participant. Using the ProNutra production sheet, document the preparation of each ingredient included in the composite. Another staff member should verify accurate production of the menu day. Fill the wash bottle to the top with distilled water. Weigh the wash bottle filled with distilled water and record the total weight on the production sheet. Please note that each menu day has a specific amount of distilled water that should be added to the blender to aid in homogenization. The chart with these amounts is in the composite MOP. Weigh the dry blender canister and lid and record the weight on the production sheet. Please refer to the composite MOP for further detail regarding the blending procedure. Once all the foods for a particular menu day are prepared, the foods must be added 1 meal at a time with weights taken in between each addition. Use a silicone spatula to capture all foods left on the plate or container. Document the weight of the food after each meal addition on the documentation form. Secure the lid and blend on the stir or low setting for 30 seconds. After that, open the lid and add approximately half the distilled water amount specified for the menu day. Then, using a timer, blend on high for 60 seconds. Once 60 seconds has elapsed, stop the blender, remove the lid, and rinse the contents of the canister sides and lid back into the homogenate using the rest of the specified amount of distilled water. Using a timer again, blend the homogenate for 240 additional seconds on high. The total time blending on high should be 5 minutes. Once the composite is fully homogenized, transfer the canister and lid to a tarred scale and record the total weight. Use an NSF-approved thermometer to take the temperature of the homogenate and record the temperature on the composite documentation form. To collect samples, return the canister to the blender and run on stir or low to ensure the homogenate does not separate. Use a pipette or turkey baster or syringe to fill both of your prepared sample bottles with 240 grams of homogenate and then place the cap securely on each container. These prepared samples should be immediately transferred to a minus 20 degrees Celsius freezer until ready for shipment. After samples are stored, complete the RedCap Diet Composites production log using the production sheet. Document the final weight of the distilled water used and the total weight of the homogenate prepared on this sheet. Lastly, scan and upload the production sheet into RedCap. Once all menu day samples are prepared and frozen, one of the sample bottles from each menu day will be shipped to the food chemistry testing lab, and one will be stored until the results are returned, which should take about 2 weeks. The results can be viewed in the RedCap Diet Composites production log. To ship samples, first ensure that they are frozen and the lids are secure. Then log into the Food Chemistry Lab website to generate the order form. Once you print the order form, place samples in a plastic, sealable bag to prepare for packaging. If you're using ice packs, place ice packs in the bottom and on the sides of the insulated box you're using to ship the samples. If you're using dry ice, place 15 to 20 pounds of dry ice in the bottom of the insulated box and separate the dry ice layer from the samples with cardboard or by wrapping the samples in bubble wrap. Make sure to include the Food Chemistry Lab order form between the insulated box and the cardboard box. Additional detail for shipping procedures can be found in the MOP.